Oh, they are not online yet. I'm sorry. They will be available later. Sorry for that. Yeah. So, is this working? Should? Yes, it is. So, brief recap on what we talked about on Tuesday. So, I was talking about 2D structure prediction and specifically about EMBER2, which is one of the methods from our chair where we're using protein language model embeddings for the prediction of distance maps. Brief recap on how that worked. So instead of creating MSAs like most state-of-the-art methods would do, we are instead using PROT5. So we're throwing the query sequence into PROT5 and extract embeddings from there. They can be either 1D or 2D. 2D is going to be the attention heads of this transformer model. And the attention heads are what we're using here for EMBER2. We're doing some pre-processing on that. And as we've seen, even from this unsupervised training of these language models, we already have kind of a structural signal in there, which is, at least for some samples, similar to what we observe in the actual contact map based on the PDB structure. So based on those attention heads, we then throw a deep residual convolutional neural network on that and predict distances, which we can then use in a separate stage for folding. Now, one of the big advantages of this is, of course, we get rid of this MSA computation part, which is where most of the compute comes in. So it's much faster to do inference with embeddings than it is to do it on evolutionary information. And the other secondary benefit from this is that we are in fact protein specific. We are not averaging over a family of proteins as you would with an MSA, and that makes systems like those more sensitive to small changes in the sequence. So for example, we could do something like mutation effect prediction, where we take a protein, we predict the structure for this wild type version of the protein as we observe it in the organism. Then we can also, since we're also so fast, produce structure predictions for every kind of point mutation you could introduce into that sequence, predict structure for those, and then see what is the difference between the predicted structure for the wild type and for the mutant. Then if we measure these structure deltas for all these mutants and then correlate that with data from deep mutational scanning, for example, we see that this correlates much better for our embedding-based approach than it does for an MSA-based system like AlphaFold. So even though the overall quality of EMBER2 is much lower than AlphaFold 2 if you want to have like a good high quality model for a protein, but if you want to introduce mutations, it's actually much better to use embedding-based systems, at least on a broader scale. Now, based on this idea, and this is what I've not shown on Tuesday, you can also do something like a mutation movie. And I'm first going to show you this in 2D, but then we're going to talk about later how we can apply this to 3D as well. So what you see here is essentially the same thing I talked about. So for one sample, we mutate every single position to every possible amino acid. And then we're just making a movie out of the individual distance map predictions that we get out of EMBER2. And this is just kind of cool to watch, to observe like how does a specific change in a sequence relate to a specific change in the pattern of the structure. And then what I'm showing here on the right side is essentially a mutation profile, where for every point mutant that we generate, we're measuring the structural difference to the wild type. And the brighter it is, like the more yellowish it is, the bigger the impact, the bigger the structural difference between the two. You see this also summed up over each column, which also tells you that, for example, for this position here at 40, where they have the biggest impact, this is actually not overall the highest, it's actually this column here. So this gives you kind of an idea like how sensitive is a certain position overall to a specific change. Yeah? We don't know. That's the thing. We don't really know. It might also be. Both could be. Yes, exactly. So we are of course not sure if what we predict is actually reflecting what is going on in reality. It could just be noise from the network, and in some cases it certainly is. In many cases it probably is. But this is exactly why we did the study where we correlated it with deep mutational scanning, so we can see there is at least some correlation. So at least we capture some aspects of this. It's not just noise. But you're absolutely right, we don't really know what we actually observe. Is this just noisy stuff that the network produces, or is it an actual change in the conformation? And this needs further validation, of course, yes.
Your point was, how does it happen in nature, not how does it happen in my prediction, right?
In nature.
So yes, you're right. So these are two possible reasons. It's much simpler. You know, remember the protein is densely packed. So if you mutate the small residue somewhere there to a larger one, you immediately get strain. As simple as that. Vice versa, if you have a large residue and you have a smaller one, you create sort of a vacuum. And ultimately, this is on top of what you said. Yes, but all of these things happen.
Yeah. And then it highly depends on which position it actually is in the protein, right? We talked about this on Tuesday as well. There are some positions where it doesn't really matter if you mutate anything because it's not so important for the formation of the protein. So nothing really matters here, whatever amino acid type you introduce. But some positions are really crucial for the fold, and if you change anything there, it will really disrupt the structure, right? Yeah. Well, this is not a cherry-picked example, it's just one example. We're looking at many more movies in the end. I hope you brought popcorn. We're watching many movies. It doesn't work for every protein like this. I mean, there are some proteins for which just the prediction is really bad, and then it doesn't make sense to really look at anything. But we're going to tackle that exactly when we're going to 3D prediction, because then we have a good reliability indicator that tells us if our prediction is valuable or not.
Again,
So,
I believe you understand the question too much on the side of the prediction.
okay.
So not every protein is as stable that no variant is going to change it. In fact, many proteins are such that a single variant will change something major. Okay? But don't forget, by changing every single position, you're still changing on one residue at a time. So we never look at two residue changes. Right? And so there is a relative stability against a single change. Again, in some proteins you will have an effect of some change of single changes. So this white, these yellow positions, there may be cases where but we don't see it in the movie. But in principle, most proteins have a sort of soft spot against some variants. And I'd say that most proteins have one variant that will sort of change something more radical. So in that sense, this is an example that is more stable than others. Yes, the moment we do two, the disruption, so the probability of disruption increases dramatically.
The problem is, I mean, I would also like to go deeper in mutation space, but then it becomes very quickly very expensive computationally, because the amount of different variants that you can have, if you're just going for two, it just explodes, right? And if you go deeper, it's even worse. So this is something I would like to do at some point, something along the lines of exploration of structure space, where you would also try to go from one structural conformation to another one. Figure out what kind of mutation path you need to actually get to a different fold. This would be very cool and maybe also very useful for protein design, but we're not quite there yet, because we need a good heuristic on how to navigate the space efficiently to not just blow up the possibilities too much. Okay. Any more questions on this before we move to 3D? All right. So the first method I'm going to talk about is AlphaFold 2, obviously. This is the big system that everyone uses to get high quality predictions nowadays. Now, before I start explaining anything about AlphaFold, first a disclaimer. AlphaFold 2 is such a complex system. The supplementaries is like 80 pages long, and this is also not covering every detail. So I can only hope to give you a broad overview of the pipeline in general, and then some specific aspects I find particularly interesting, but this is by no means exhaustive, right? So if you need more information, just refer to the paper, the supplementaries, and the like of blog entries of people trying to dig into the system and understand how it works. So before we talk about the pipeline itself, first some terminology, because there are a couple of systems available at this point you could use. So first off, there is, of course, the official AlphaFold implementation. This is the inference pipeline they push to GitHub. Actually, for quite a while in the field, we were wondering if they would actually publish anything, because ultimately Google and DeepMind are not public research institutions. It's a private company. They are mostly concerned about shareholder value. So we thought maybe they would monetize the system in some way, but actually they did push this out, at least the inference part, but they did not publish the training code. So, but this is also something which is quite common in machine learning. Most people publish the inference code, but not necessarily the training code. And I believe this is something we should change because especially for young researchers, it's much better to have something to work with and actually get into the code, understand how the training works. And if you did any kind of machine learning, you also know it's actually quite hard, especially for a sophisticated system, to infer the training procedure just from the inference code. There's usually so much stuff going on behind the scenes that it's really hard to reproduce that. So yeah, so you have the official inference pipeline from AlphaFold, but if you want to run AlphaFold predictions, I would actually suggest that you use a different system, and that is CoLabFold. CoLabFold is an effort of quite a few people in the field of structure prediction, trying to provide like a general protein folding platform on Google Colab. And this is not only offering AlphaFold 2 predictions, it's also offering Rosetta Fold, and I think now just very recently also OpenFold, which I'm going to talk about. And the main benefit of this approach is that they exchange the MSA generation procedure, and instead of using HH-Blitz, which is what AlphaFold uses, they're using MM62, which is just a lot faster. So this speeds up the whole prediction procedure quite a bit. Then the third one you could use, which is not so useful for predictions, but if you want to really get into the training code, is OpenFold. OpenFold is a huge effort, spearheaded by Mohamed Al-Quraishi, who's a well-known guy in the field. And he and his team attempted to reproduce essentially AlphaFold as a whole, as a trainable system. And they're mostly done with this at this point. There was just a Twitter post from Mohamed a couple days ago mentioning that they're now at the point where they actually slightly outperform the official AlphaFold 2 version with their OpenFold implementation. So we're at a point where we can really do this with publicly available code, which is quite cool. Now, let's go back to the story on how we first got to know about AlphaFold 2. So this was in December 2020, after CASP 14. And we didn't even know at this point whether or not DeepMind would participate again. And we were all waiting for the results on 3D structure prediction, like who's going to come out on top. This time around, we knew that AlphaFold 1 won the previous CASP competition, CASP 13, in 2018 by a smaller margin to the next group. And then early December, we got this. This was the first graphic published by the CASP organizers, and everyone was so shocked at what they saw here, because this has never happened. This has never happened. So this is a performance metric that you see, and these are all the participating groups in CASP 14. And as you can see, one group just totally outcompeted everyone else. They were just so much better. And then some groups coming here, which are kind of good, and then it kind of trails off, which is what you normally see. So starting from second group, this is usually the picture you get in CASP. If one group's being pretty good, and then some others also kind of competing with that, and then it trails off kind of linearly. These are often just old methods, which are just handing in predictions because they can, which is often a good idea because that allows us to see the progress over years through better machine learning techniques or just growth of structured databases. But this is a margin we have never seen. And of course, it was quickly then known that this was actually AlphaFold 2. And obviously, this changed the field quite significantly. So how do they do this? Yes? CoLabFold didn't exist at this point. We look at a graph later where I compare performance with also a bit of CoLabFold, but at this point, none of that existed, right? This was just the first day AlphaFold was visible, essentially. So how do they do this? We're now progressively getting deeper into systems of AlphaFold, and this is kind of the most broad overview that you could have of AlphaFold 2. So let's walk through this. The data flow direction, as you can see, is from left to right. So we're starting with an input sequence here. This is the original query sequence from a FASTA file. And then we're doing a couple of things. Now, obviously, we have this MSA generation procedure where we're looking through large sequence databases like BFD, metagenomics databases, look for related proteins in other organisms, build up the MSA. Also building up pairwise information, and this is mostly fed by structured database search. So AlphaFold also looks in structured databases, PDB, and looks for templates. So essentially looking for homology modeling approaches. What it does is, well, it collects distance maps for all of the fragments it could align with known templates and puts it into the initial pairwise representations. Then we're getting into the main backbone of the network, and this consists of two major parts. We have the evoformer blocks, and we have the structure modules. Evoformer blocks are the largest portion where most of the free parameters are in. These are 48 blocks they use in their final architecture, where they put in 1D information. We could call this, well, there are multiple terminologies for this. They call it MSA representations, we could call it 1D representations, or I would also call it node representations, because ultimately we can see this as a graph inference problem. So this is the 1D part. Then this we can consider the 2D part, or pair representations, or in graph terminology, edge representations, because they contain information of residue pairs, right? So we have this 1D and 2D information which is being pushed through the network for each evoformer block. And then after these 48 blocks, we have 8 blocks of structure modules where we introduce 3D information. So for all of these blocks, we're just having 1D and 2D representations. Only here 3D comes in. We're going to dig into that, how that actually works. And that is then creating the final 3D structure in the end, which is an all-atom model. It's not just backbone, it's actually predicting coordinates for all heavy atoms, so everything excluding hydrogen atoms. Now, the other thing that AlphaFold does, and which is kind of a novelty in the field, no one has done this in this way before, it's also predicting per residue how confident this prediction is. And this is what they measure in p-LDDT. That's the predicted LDDT, which is a local distance measurement. It essentially tells you in a sphere of 15 angstrom around a residue how conserved compared to the ground truth structure is this local environment. They do this for every residue, so what you see here is in terms of color coding, the blue is the most confident one, and the more it goes into the greenish, yellowish, reddish spectrum, the worse the confidence is. And what they could show is that this highly correlates with the actual performance. So it's a very reliable reliability indicator. Yes? Sorry? Conserved means, in more detail, means that it looks for the fraction of contacts in that sphere which are present in the prediction as well as in the ground truth. So essentially what you're doing is you're looking at this sphere of 15 angstrom around a residue. You're looking at all the other residues which are in there at certain distances in the ground truth structure. Then you're looking at the prediction around the same residue, and you simply count, essentially you count, it's a bit more complicated, essentially you count the fraction of residues which are also there, which should be there, right, in the sphere. Kind of like that. It's a bit more complicated. Look it up, LDDT, there's a bit more involved in that, but essentially that's what it is. Yeah. It does this at different thresholds, different angstrom levels, essentially, and then kind of sums this up and averages how many you get. Yeah, this is kind of an arbitrary definition of a contact that just happened to be like that, and the cusp, we had that in cusp. I think it was useful for contact prediction back in the days at some point, but for LDDT it doesn't really have anything to do with the contact definition. It's just useful to use 15 angstrom, and this is also kind of arbitrary. You could make it larger or smaller, but it's just it's a reasonable distance. Okay, so the cool thing is now we have these predicted confidence values, and as you can see, this can differ in a protein. So some parts of a protein can be predicted quite confidently, and other parts less confidently. And again, as AlphaFold, as DeepMind has shown, these confidence scores are actually quite reliable. So whenever the confidence is really high, we can be pretty sure that the structure at this point is correct, and vice versa. Now, what they could also show, yes? Yes.
Is there a correlation between, because the inside we have more relation to the other part of the protein and the outside of it, like we have no connection or less connection?
There's a clear correlation. This was what I was about to say. Yes, there's a clear correlation between disorder in a protein and low confidence scores, which kind of makes sense, right? Because in the structures AlphaFold is trained on, there's more uncertainty in the areas in a protein which are disordered, right? There's also more experimental error, so the network cannot quite learn how a correct structure would look like. And there is, in fact, no correct structure. If it's disordered, it can be flexible, right? So yes, we see a pretty good correlation between disorder and predicted LDDT, which is useful in its own way because you can treat it as a disorder predictor, right? Yes? So what they're doing as well, in order to make the network more powerful, is they're not stopping after one pass through this pipeline. Instead, what they're doing is they're taking the 1D and 2D representations and also structural information, and then they feed this back into the inputs, original inputs. They concatenate this and then feed it through the network again. They actually do this multiple times. You can control how often you do that. In the end, they did it 3 times usually. And what this does is a couple of things. So first, it makes the network deeper, right, without introducing more free parameters, because they're reusing the same blocks for this, okay? But then also, it kind of trains the network to do iterative refinement. It learns that as the input, it gets some kind of a rough estimate of the structure, and it learns how it should optimize that to make it more accurate, right? So this is the cool thing about it. And you can cycle through this as long as you want until you get a high predicted confidence. So this is another application for this confidence measure. If you can estimate yourself in your predictor how good your prediction is, you can control how often you recycle. And this is what they do as well. So you just iterate until your predicted confidence does not improve anymore, and then you stop. And then hopefully you have the best possible structure. Okay, so let's zoom into one of the core components of the network, which is the evoformer block. And that looks like this. Looks a bit intimidating. I'm going to go through this on kind of a high level. So again, we're having 1D information and 2D information coming in, and also 1D information and 2D information coming out. And then it goes into the next block, right? We have 48 of those. There are no shared weights between them. So these are 48 individual blocks with their own weights, which are trained individually, well, together in one system, of course. And what we see here is that we first have computation on the 1D part and then a computation on the 2D part. Now, the 1D part is relatively simple. It is essentially row-wise and column-wise self-attention, but there's also a bias introduced from the pair representation to kind of feedback information from the 2D part into the 1D part. But it's a relatively weak coupling, and this is kind of a common theme you will see here in AlphaFold, that the core of the information, the most important information, is actually in the 1D part, not in the 2D part. The 2D part is kind of like a memory system to aid the 1D part, but especially in the structure module later, the 1D part is essentially what the network uses to build the 3D conformation. Now you have these row-wise and column-wise attention, and then it's gonna transform, it's gonna let the information flow into the 2D part by just taking the outer product of the 1D representation. That gives you a pairwise representation, right? This is what you add into the 2D information that you got in the input. And now there are a couple of triangle updates happening here. And this is, I think, one of the core ideas of AlphaFold, which are interesting. Now, in order to explain that, let's look at the other parts of this figure here. So if you consider a pairwise representation, you can think of this like a contact map or a distance map, right? It's not exactly like that. It's some latent space representation, of course, in this network, but it's something like that. It's some kind of relationship information between individual nodes. So if you consider this as a graph, you have individual nodes, which are residues, and then you have these edges, the relations between those going back and forth. Now, when it comes to distance maps, for example, there's one thing you need to consider. Now, a realistic distance map has some constraints on how it can look like. For example, it has to fulfill the triangle inequality. This is immediately apparent. So if you have 3 points in 3D space, and you have these distances between those points, and let's say we call this is A, this is B, and this is C, then you know that C has to be smaller or equal than A plus B. It cannot be larger than that, right? So this is a constraint that needs to be fulfilled in a distance map as well. But so far in every other prediction system that predicts distances, it was not enforced. And what you could get out as a distance map could be anything that violates that. But it would be important to somehow consider this. Now, what AlphaFold does, it's not actually enforcing this, but what it does is it kind of gives the informational way for the model to learn something about this. And this, I think, is a cool concept. And this is what they're doing with these triangle updates. So what they're doing is to update these pair representations is they're having multiple steps where they let information from neighboring edges flow into the update procedure for that specific edge we're currently looking at. So kind of taking pairs of pairwise representations for this. They're doing this in a couple of different ways. So they're having multiplicative updates using the outgoing edges and incoming edges. And then they're having also self-attention on edges coming from the starting node and from the ending node. And so this gives the network the ability to learn something like respecting triangle inequality without actually forcing it to do so. This is kind of in the mindset of machine learning. We're not explicitly encoding constraints in there, but we allow the network to learn them efficiently. Questions about this? Okay, so let's take a quick look at the structure module. Again, we quickly go back. So we talked about the evoformer block here, which just computes between 1D and 2D. Now we have the structure module, which is at the end of the pipeline. And this looks like this. Now what we feed in here, and which is the most important part, is a single 1D representation. This is kind of the first row of the MSA representation that we had after all these evoformer blocks. And this is really at the core of the construction of the 3D coordinates then. We also have the pair representations going in, but really, at least as they claim, the core information is in the 1D representation. Now, how do they do this? They model backbone frames. And backbone frames are essentially just these rigid body frames which are representing a residue. And how they do this is they represent this residue just by one translation and one rotation. So essentially, there's some position where it is in 3D space, and there's some rotational alignment for the residue. And this is essentially all they do at this stage of the structure module. Now, initially, these coordinates and rotations are at the origin, and the rotation is just identity. And then they update those relative rotations and translations in the structure module. Through this module here, we're not going to dig into the details of that because this is going to become very math heavy. This is the invariant point attention module, which essentially, just on a high level, does a transformation with this, which is not dependent on the rotation of the global scale of the protein, right? And this is important because a protein is still the same protein after you globally rotate or translate it. Right? It's still the same thing. There should not be any difference, and the machine learning system should not care about that. And this is exactly what they realize in there. This is what we either call invariance, if you have a feature that simply does not depend on where it is. This is something, I think I had these slides in here. Yes, so let's consider something like a CNN. In a CNN, you have translational invariance. So if you want to detect an object in an image, like this cat here, the convolutional kernels don't care where the cat appears in the image, if it is here or there. It doesn't matter, it will still detect it because we're running the convolution kernel along the whole image, and well, wherever it is, it's going to detect the signal that it's trained for. However, they are not invariant to rotation and scale, right? The convolutional neural network. It might still detect the cat because the cat has some features which are implicitly rotationally invariant, like the eyes of the cat, because they're round, it doesn't matter how you rotate them. But overall, if the system is kind of trained on recognizing certain alignments of structural elements in the face of the cat, it would not just naturally pick this up. We would have to actually train it to do that, right? So it's important for the structure module here in this invariant point attention module to not care about the actual rotation on the global scale. If you're interested in the math, look in the supplementaries. There's a great explanation on how it works on a more technical level. So again, it then updates these relative rotations and translations and kind of builds the backbone structure, but again, it's not actually enforcing any peptide bonds. It's treating the residues, as they say, as a residue gas. It's just individual rigid body frames, which can essentially take any kind of position in space, but through the loss functions, we're going to talk about that, it still works out and it kind of aligns in a correct way to form a correct structure. And then at a later stage, at the refinement stage of the network, they actually do have a loss which enforces peptide bonds and correct distances between atoms, but this comes at a later stage. Okay, talking about loss functions. Now, usually in a machine learning systems, the ones you've probably worked on at some point, you have like one loss function. AlphaFold has a lot. It's not just one, it's many. So, and this is also why its system is so powerful. They're just enforcing so many different constraints that it really learns to represent structure well. So what they're doing is they are also doing 2D predictions. So they have a 2D histogram loss in the same way we talked about on Tuesday. They have an interesting loss, which is experimentally resolved loss. So they're predicting in the system if a certain residue is actually resolved in a structure, which is a cool thing. I didn't think about that. Then they have the most important 3D loss, which is the FAPE loss. This is the frame-aligned prediction error. And this is essentially taking taking these frames that we talked about, these backbone frames, and again looking at a local environment and all kinds of different alignments that you could have with the ground truth structure, then computing essentially something like an RMSD from that. Then another cool thing they do is called the masked MSA loss. They're kind of introducing a transformer concept here. So they're randomly masking out certain positions in the MSA and then trying to predict whatever is there, which apparently helps them. They also have angle losses in there, so they're predicting torsion angles from the backbone, which helps the reconstruction. And as I said, they're predicting their own performance. So they predict their LDDT, and therefore also they have an LDDT loss in there. They have also another metric, TM score. This is a global distance metric, whereas LDDT is local, TM is global, so it needs a structural alignment. They also have an output unit in their system which can predict the TM score, which you can switch on or off in the training if you want. And then again, for the most part, they are training without any actual enforcement of peptide bonds or atom distances. They do this at a later stage. So they first train just as it is, and then in a refinement stage, they actually activate this violation loss to enforce peptide bonds and so on. But again, this happens only during fine-tuning. They claim in the paper that if they were to enable this early on during training, when you start from scratch, then it's just making training dynamics very shaky. So this is something you have to do after the network has kind of learned how to position these rigid body frames in space. Yep. Slide 10. Yeah. We don't know what it is. I mean, I was just saying you can think about it like a distance, but it is just some kind of relationship information between these two positions, whatever it actually is. I mean, what the network encodes, we don't quite know. It is some kind of latent representation of a relationship between those positions, whatever it actually is. It's not necessarily doing that. This was just to motivate why they are coming to this kind of update scheme. It's just that the network has the ability to infer something about the relationship of those edges. This is all we do. We never enforce anything specifically, right? We just give the network the ability to do so on its own. Kind of like we always do in machine learning, right? We never explicitly, or we almost never explicitly encode, we just train the system in some kind of interesting way. Okay. So I think one important lesson learned from AlphaFold 2 is really that just doing a lot of small things right goes a long way. So it is absolutely true, and I think this is a life lesson, really. I mean, we always worry about these big things like finishing our degree with a good grade, or finding the best job available, or finding like the best relationship partner, whatever. I mean, these are important goals, and you should pursue them in some way or another. But it's also important to note that if you just do a couple of small things right, this really moves you forward, especially the things you do like every day. This is super important because you spend a ton of time on that, so think about that. So anyway, back to AlphaFold. Here's a big ablation study that they did. They probably did a lot more, but I mean, it's just, there's so many knobs to turn and so many things to leave out and see how you would perform. So what you see here is the baseline performance of the model as they introduced. Self-distillation training, I think I had a slide. Oh yeah, this is coming. So yeah, I'm going to talk about that in the next slide anyway. So you see a couple of measurements here for other training runs where they simply left out certain aspects of their pipeline. And as you can see, most of them individually do not really contribute that much. It's just a small change. It's just a small little bit, and you could say, yeah, well, do I really have to go through the effort of implementing that? It's a lot of code I have to write and test and debug. Can introduce some mistakes. But, well, they did such a good engineering job that it actually worked out, and it did not degrade performance through bugs, but it actually all just added up to this pretty decent performance here. So I think this is just an important lesson learned, right? Do the small things right. So a few words on data, because I also believe this is very interesting what they did here. Now, normally when you're training a structure prediction system, you will cluster your training set, right? You don't necessarily have to do that, but it's a pretty good idea for two reasons. The first reason is you just want to bring the size down so your epoch is a little bit shorter, right? Because you will have a lot of redundancy in there. Why train on very redundant proteins? It's just best to take representatives, cluster representatives, just makes it faster. But also, there's the thing, if you take PDB, for example, as your training set, you will have a bias towards large families in there. For some proteins, you have a lot of structural data in PDB, and for some protein families, just very few, right? Or maybe just one example. Now, if you were to just train on the full redundant thing, you would certainly have a bias in your structure predictor for these large families because you're kind of overtraining on those, right? So this is something you want to avoid. Now, what AlphaFold does is they're not completely throwing out everything else than the cluster representatives. So they are clustering by 40% sequence identity, but they're keeping all the sequences, and then during training, they're randomly sampling sequences from their training set with a probability inverse to the size of the cluster that they're in. So if you have a sequence from a cluster where there's only like a handful of sequences, then the probability of sampling that during training is much higher than having a sequence which is part of a larger family. You usually do not sample that often. So that's one of the ideas. That seems a bit too complex. You could also think about just essentially during training, randomly sampling one sequence from each cluster. This is a deal that RosettaFold then does. Similar concept. Now, another thing they do is what's called self-distillation. So they're not training like the finished AlphaFold 2 thing from scratch to the final version they publish. Instead, what they did is they trained a preliminary model on PDB structures, just as you would normally when you train a structure prediction system. Then they took this preliminary model and they made predictions for hundreds of thousands of structures, in this case from Munich class 30 to have some kind of redundancy reduction in there. And then they took the most confident predictions of those and then trained their actual AlphaFold 2 model. And on a mix where there's only a quarter of actual PDB structures in there, and 3/4 were just these highly confident predictions. So they had a much, much larger training set to work with, and this worked quite well. As you can see here, this is probably one of the biggest boosts from baseline to self-distillation. That's probably worth more than many of the other concepts here, and it's pretty easy to do. You don't have to implement much for this, right? Okay, so much about AlphaFold 2. Any more questions on this? Again, if you want more information, there's so much else to learn about it, but you would have to look at the supplementaries or blog posts, etc. Okay? Okay, let's quickly talk about UFO-fold. UFO as in flying saucer fold. This is work from Michael Jaumann and Duc An, which are working in our labs. These are two students. Michael Jaumann just handed in his master thesis, and essentially UFO-fold is the work of this master thesis. Now, what they did is they took OpenFold, since we have such a almost complete system which faithfully constructs AlphaFold 2, they could take OpenFold and just replace the whole input part. They were just throwing out template, search, and MSA generation and plopped in Prot-T5. So everything we do now is we put in sequence, put it into Prot-T5, extract 1D embeddings, and push that into the MSA representation, which no longer is an MSA representation at this point, it's just the 1D embedding from Prot-T5. And instead of template information, we're putting in the 2D attention heads into the pairwise representations. And everything else is essentially the same. The rest is the AlphaFold architecture, just retrained now on Prot-T5 embeddings. Now, this seems like a small feat, but it's not, because AlphaFold, or OpenFold in this case, is a pretty complex system, and just plugging out the whole input part and doing something else with that was actually a major effort. So this took a master thesis to complete, and Duc An is now taking over while Michael Jaumann is trying to write this up into a paper, and hopefully we have a preprint in the coming weeks on this. More on how it performs and compares later on. Let's first look at RosettaFold. This is the other big system that we currently have. So the first question is, why even look at RosettaFold, since AlphaFold 2 is obviously the best system by far? So why should we take a look at it? There are a couple of reasons for this. The first one is, this is from Baker Lab. David Baker is one of the guys who has been around in the field for a long time. His lab has been working on structure prediction for, I think, decades at this point. So it's always interesting to see what they're coming up with. The other reason is it's an end-to-end prediction system, or at least potentially it can be, which also goes immediately to 3D coordinates, and this is something that is still pretty rare in the field. And then there's simply the fact that even though AlphaFold 2 is a lot better than RosettaFold, RosettaFold is still a lot better than everyone else. So this warrants also a closer look at it. You see a bit of the performance here. This is different implementations essentially of AlphaFold. So here's the comparison with CoLabFold, by the way. So the original AlphaFold, how it is in CoLab, and then other implementations, and then RosettaFold, which is the purple upside down triangle. So as you can see, these triangles are consistently below the AlphaFold curves, especially here for those samples. But still, what you have to keep in mind is for the TM score, a TM score above 0.5 generally means that you capture the idea, the structural idea of the protein you're doing your prediction for. And everything above 0.7 is a pretty good match of the structure, and everything else is then just cherry on top and getting it like totally right in terms of atom positions. And if you keep that in mind, you can see that for most of these predictions here in CUSP14, Rosetta Fold is performing pretty well, so it's getting the structure generally right. So there are a couple good reasons why it still warrants a closer look. The other one is more of a practical concern. If you try to get into AlphaFold or OpenFold, this is a pretty big task. To really understanding that code and in order to change something in there is quite challenging. And this is not quite the case for Rosetta Fold because it is a much simpler architecture, and the code is really well written and accessible. I was really amazed how well this was done. And this is largely thanks to Mi-Kyung Bak, who has been the first author on Rosetta Fold. She's a postdoc in Baker Lab, and because of her effort, it's quite accessible. And this is not quite the case for OpenFold. So quickly on the data set that I've been using, they've used a PDB snapshot from 2020. They also do redundancy reduction with 30% sequence identity, getting out 23,000 clusters. And kind of similar to AlphaFold, they're not throwing away anything that is not a cluster representative, but they're also just selecting a random protein from each cluster per epoch. So eventually they're going through all the training data they have. They just make sure to remove the bias towards large families, which is the main reason why we do the clustering in the first place. They also do some MSA subsampling, but this is kind of what every MSA-based method does nowadays. So a very general overview of the architecture of Rosetta Fold, and you see immediately that there are some conceptual similarities to AlphaFold 2. Now, the interesting thing is by the time that Rosetta Fold was published, AlphaFold was already out, but there was not that much information available. So this is work that has been done in kind of the first half of last year. So remember, we had AlphaFold 2 being visible in December 2020, and then everyone made a big effort to try to gain as much information from what we knew about AlphaFold 2, and this is probably the best effort so far in a different system. So you see, we also have this kind of this 1D, 2D block scheme that we also saw in AlphaFold. So we're processing 1D and 2D together. And then we have kind of the equivalent of the structure module from AlphaFold here in this 3-track pipeline, where we introduce 3D information and then have information sharing between all of those stages, right? Otherwise, it's a pretty similar system. So we're throwing in an MSA representation directly, not just some evolutionary coupling scores. And we're throwing in template information again from the searches in structure databases. I have to hurry up. Oh man, okay, it's already 1:15. So what comes out are a couple of different representations. We have 1D representations, but even more importantly, we have edge representations, like the 2D features. We have distances and angle maps. And this is a pretty big system. So it's in the same ballpark in terms of 3 parameters as AlphaFold 2 with 130 million. And the loss functions used are pretty simple, though, compared to AlphaFold 2. So it's a bit more to what you are maybe used to. So it's 2D distance and angle classification with cross entropy. Then it's simply RMSD for the 3D distances. And we also have this predicted LDDT based on C alpha, just like AlphaFold 2 did, because that worked so well, and it also works pretty well for Rosetta Fold. Now, this is what they published in their paper. This looks a bit more complicated. This is why I showed you the kind of reduced version before. This has a bit more information in there. Same idea, though. This is the 2-track block here with 1D and 2D information. This is the one with 1D, 2D, and 3D. And you see some of the actual transformation steps. It's mostly based on attention between 1D and 1D and 2D and 2D, and also when you feed back 2D information into 1D, it's an attention-based mechanism. What is interesting is how they actually handled the 3D part, and this is what we're going to take a look at now. Or rather, let's first look at the blocks themselves, how the information passing actually works. So similarly to AlphaFold 2, we first have an update on the 1D part, then the 1D information is propagated into the 2D track. We have the pairwise update coming, and then the pair information is transported back into the 1D part. After that, we have the actual 3D update of 3D coordinates, which is making use of the 1D and the 2D information. and then afterwards projecting back into the 1D space, into the MSA space, which is also kind of in line with the idea from AlphaFold 2. Remember, they claim that most of the important information is actually in the 1D part, not in the 2D part. And this is what RosettaFold converged to as well, so this is why they're updating the MSA part and not the pairwise representations at this point. Now, in terms of the 3D parts, there are again two. One is for the initial generation of coordinates, and the other one is for the update of coordinates. And they work in quite a different way. So for this one here, for the generation of initial coordinates, we're using a graph transformer on a fully connected graph, which is kind of one of the drawbacks of the system, because a fully connected graph for a large protein can be quite big, right? So we're throwing in all the MSA features, the 1D information we have, and the pair features, the 2D information, which gives us the edge representations, do a graph transformer block, and what we get out are backbone coordinates for the N, for the CA, and for the C. Now, in the structure update part, the cool thing to note about this is that we're not actually predicting new coordinates, but what this whole thing does is it computes displacement vectors. So it's computing a correction for the coordinates that were coming in. And the new coordinates are then the old ones, which were coming in, plus the displacement that we computed. And this is an interesting point, because that means it's refining coordinates. It's not doing something new, it's just refining them or learning to refine them. In the interest of time, I'm not going to go into too much detail on that or that. So just in terms of results, how does it look like? So RosettaFold is actually, in the way it's published, not a system that goes immediately to 3D, because that actually performs a little bit worse. So they have two versions. They have the version which is end-to-end, predicting backbone coordinates, and they have the version which uses pi-Rosetta folding, where they use the 2D information they predict, so essentially distance maps, then have the separate folding stage like we had for regular 2D predictors we talked about on Tuesday. And that is actually the best performing one they have. And again, this is still falling short of AlphaFold 2, but still pretty high performance. Yes? It is essentially the same thing as the invariant point attention, or kind of the same thing conceptually as the invariant point attention in AlphaFold 2. So it's a transformer which has the same property of rotational equivariance. So whatever the rotation is, it doesn't really care, right? This is the idea. So no matter how the 3D point cloud is rotated, it's still doing the same kind of transformations with this. Sorry? What kind of twist? What do you mean? Mm-hmm. Mm. Mm. I'm not exactly sure. You have to look into the details of the SC3 transform. I'm also not quite familiar with that. I'm sorry. Yeah. Okay. Other questions? Other comments?
So I'm not entirely sure what you mean by twist, but twist is not a very likely event. So the protein has a lot of flexibility, that's true. But don't think about it like a thread that you can just twist. Because the way you connect it, you have resonance, right? And they're on a peptide bond. The peptide bond has only a certain rotation. So this I cannot answer your question better, but the idea of a twist is twisted.
Yeah, I mean, if that happens just on the level of prediction, the SC3 transformer should pick this up, right? Because this is not just a global transformation, it's actually a transformation of local parts, right? So that's not the same. So it should recognize that, yes. Okay, let's briefly talk about Ember 3D. Ember 3D is my take on this, and this is more based on RosettaFold, whereas UFO-fold from Michael Jaumann is close to OpenFold, obviously. Ember 3D is largely inspired by RosettaFold. So again, looking at RosettaFold as the full architecture, I was trying to make this much, much faster and leaner and smaller, because this is kind of my theme. I want to make it quicker. So I boiled it down to just the essential components, basically, so just the initial 3D generation and one of these 3-track blocks, and then again also feeding in Prot-T5 information instead of any kind of MSA-based information. Otherwise, it's essentially the same thing in terms of output and losses, but as you can see, it's a much smaller system with 3.3 million parameters against 130 million. Now, in the meantime, with some more experimentation, I've extended it slightly, and I saw that it's actually beneficial to include at least one 2-track block and one more 3-track block, but it's only adding a few more free parameters. Now it's at 4.6 million, and it gives slightly better 3D performance, but essentially same thing. Some other modifications I did with the system were that I completely reworked the 3D track and included an NVIDIA SE3 transformer. The original implementation from Rosetta Fold has like the first published version of an SE3 transformer, and this is just giving better memory profile and much higher speed. So this was just a good thing to do on the engineering front. And I attempted to tinker with the architecture a bit more because I thought that the embeddings would conceptually work a little bit different than the MSAs. I thought there would not be that much emphasis on the 1D information in the system as there would be for an MSA-based system. So I tried to completely remove the 1D track, which did not work well. I tried to take the full architecture with the 1D track and just zero out the 1D input to figure out if that's important or not. And actually it is important, so the 1D embeddings are also kind of crucial. And then I tried to also tinker with the information sharing between these different tracks, right? So I thought it would maybe be beneficial to put the 3D information in the 2D stage instead of putting it back into the 1D stage, but it also didn't quite work. So as you can see, Rosetta Fold is a pretty solid system, and it's not worth tinkering with it too much. Anyway, so let's go for a comparison of these systems. So here you see all these introduced methods side by side, plus even a few more here. So in terms of TM score, you see, this is on CUSP 13 and 14, by the way, see AlphaFold and Rosetta Fold are performing quite well. For this specific set, actually, AlphaFold is slightly worse than Rosetta Fold, but this could have to do with the fact that Rosetta Fold might have been retrained in the meantime. This is from their server, so this might be one reason. Then you see UFO Fold, which again is the OpenFold version of AlphaFold 2 retrained on Prot-T5 embeddings. And then the Ember systems from me, Ember 2, which is the 2D predictor, and then Ember 3D, the newer version, which is currently in development. So a couple observations on the averages. Again, these two are of course on top. UFO Fold being essentially AlphaFold is kind of in the middle. Then Ember performs the worst, even though there's a nice step up for Ember 3D, which I'm quite happy about. And if you look at the per sample comparisons here, this is ordered by confidence of, rather performance of AlphaFold 2. You see that for most cases we have like this clear order where UFO Fold is usually the better embedding-based predictor, followed by Ember 3D and then Ember 2D. There are even some samples where we're actually outperforming AlphaFold 2. Now, the main point again is with the embedding-based approaches, we are not necessarily trying to get to the same level of quality. So we're not quite up there, but it doesn't matter because we're just so much faster. And this gives us opportunities for other use cases. So here you see some runtimes measured in seconds for two sets. This is the DMS5 set, deep mutational scanning set I was talking about for these 5 proteins for which we tried every type of point mutation. As you can see for AlphaFold 2 via CollabFold, this is taking weeks, whereas we are much faster. And you can see there's even a pretty significant difference between UFO Fold and Ember 3D. So essentially every gain in quality of your prediction you buy with an increase in computation time. So you kind of have different usage scenarios for all of them. So for the highest quality you go for AlphaFold 2, obviously. For faster speed you go to Ember 3D, and if you want to compromise where you have a very speedy prediction but higher quality than Ember 3D, then you go for UFO Fold. There was a question? Yeah? Okay, yeah. Singer stability is another data set I've been working on. I'm not going to go into too much detail, but it's a pretty big set with 200,000 sequences, where you can see that runtime for AlphaFold doesn't even make sense. It would take just weeks to compute. So we're not even attempting to do that. Now, a point about predicting one's own performance, which I think is crucial to make here. Now, AlphaFold was the first one to do it. Rosetta Fold followed up, did it as well. I'm doing it as well in Ember 3D, and well, this is actually the thing the network learned the fastest. So here you see the correlation between the true LDDT and the predicted LDDT that my system generates, and you see this matches up quite nicely. So it's a quite reliable performance indicator, which is great to have. But then I was also forming this hypothesis that maybe by predicting its own quality, it's actually learning something that might help the 3D prediction in general. Because what the network has to do is it has to kind of infer the physical correctness of each local environment of each residue, right? And if it's learning something about the physical correctness, then maybe that also helps the network in general. So I was training essentially the same system, the same architecture, same hyperparameters, same data set, and so on. And just one where I have the predicted LDDT and one where I simply zeroed out the loss. As you can see, there's a striking difference in the performance of both of these systems, right? So this is about a standard error that we gain just by predicting one's own performance. So this is something I would make as a general point. I would certainly suggest that in whatever machine learning project that you have, just try it out. Just try to predict your own performance, whatever that performance is. You will have some kind of metric that you're looking at primarily, right? Just try to predict that with your system and then see if, well, first, if it is a reliable performance indicator. If it is great, then you have that. But it might even improve your prediction performance overall. Now, this is certainly not true for any case. In the case of structure prediction, you could make the point that it kind of makes intuitive physical sense that it boosts the performance. For a more abstract prediction scenario, if you're going for like a protein function prediction, it might not be mappable to that easily, but still it's something worth trying because it's relatively easy to do, right? You're computing your metrics anyway, so why not just try it? So I hope you brought some popcorn because at the end we'll have a couple movies to watch. So this is just an interesting thing I did during training. So I tried during the development of Ember 3D to do an overfit test on just one sample to figure out how does it actually learn the structure? What happens? What does it learn first? Does it learn local conformations first, or does it learn something global first? By the way, you see the ground truth structure up there and how the performance metrics develop over the different iterations. You see how it slowly converges to this 3D conformation here. By the way, it's not rendering the secondary structure annotations. This is simply because this is just backbone, and PyMo doesn't want to render secondary structure annotations if it's just backbone. But a couple of interesting things about this is that, as you can see here, it's pretty quickly getting to this representation of these two subunits, right? This is what I found very interesting because I thought it would first go for local structural elements like secondary structure and fold those first, that maybe helices would appear first. But actually what it does is it separates those two subunits first, which I found quite interesting. This might have to do with the fact that it's just overfitting on one sample. It might look different if we're training on a couple more, but it's still an interesting observation I wanted to show you. So let's go at the end into mutation effect prediction. So again, we are so fast with systems like Ember2 and Ember3D that we can predict effects of every point mutation in a protein, if it's not too large, in a relatively short amount of time. I think I'm going to spare you that in the interest of time since we're already above the hour. Instead, I want to show you a couple more movies. I showed you initially at the beginning of this lecture how it would look like for a distance map, but it's of course much cooler if you can actually do this in 3D. So this is a mock-up I did, which shows this for one protein. So what you're seeing here are the point mutations, and it's 19 frames per second, so you see exactly all mutations for one residue per second. The position which is currently being mutated is indicated in blue. It's moving along the sequence here. You see in the color the predicted confidence of the model. So the more greenish bluish it is, the higher the confidence, the higher the quality of the prediction, and the more yellowish reddish it is, the more unsure the network is that this is correct. And then you see also the 2D distances alongside, which is of course synchronized with what we see in 3D. And you see an overall mutation matrix, which shows you for every position where are the ones with the highest impact in structure, and you're just seeing those. So the wobbling you saw are exactly those positions where we have a high predicted effect of that mutation. So this is something we want to push out pretty soon. We probably want to push this not just as a movie generation tool, but rather as a web interface, because I mean, realistically, as a user, you're probably most interested in the most impactful mutations. So it would be cool to have a web interface where you can just click on those squares and then immediately zoom to that mutation. So this is hopefully something we get into bio-embeddings, which is a system mostly maintained by Christian Delago. We're currently on the way to do that. A couple other examples I want to show you, just because I really love these movies. Sorry, I have to go through this now. So this is another example here from an E. coli protein. I can also see that, again, the confidence can be quite different in a single structure. So there's this kind of tail end. This is often what you see, that kind of a tail section of the protein is not predicted very confidently. This is also true for AlphaFold. And this is one where you also see some distinct positions where mutations seem to have a big impact. Now, of course, it's always hard to assess this. We either need demutational scanning data or some larger stability assessment of proteins to actually correlate this with experimental data, but I think it's still an interesting thing to see. Show you maybe one more. Let's see what else we have. Is this the interesting one? This is probably one where just the prediction quality is not quite up to it, but I don't know. Maybe it is a protein which is just less stable and more mutations actually disrupt the structure quite a bit. Not quite sure. I mean, the predicted structure is actually fairly close to what we see in PDB, but I think it is a bit too variable. I don't know. It seems to be very unstable. And again, it's hard to say if this is actually reflecting something that is true in nature or some artifact of the predictor. Still something we need to work on. Let's see, maybe another one. Which one was this? This is, for example, a pretty boring one. So this seems to be a stable protein. There's not much going on in terms of mutation. There's just one position near the end where it's having a bit of a bigger impact, but usually here point mutations are not doing much to the overall conformation of the protein. So this can really vary. Do I have here? Yeah, this is another case where there are specific positions where there are larger impacts predicted. But again, here overall the quality is not quite that high, so hard to say if this is actually reflecting something that's happening in reality. And I think with this I'm going to close. So thank you very much, and good luck for the exam. Questions? Then I wish you a great day. See you.